Welcome to part 2 of the molecular basis of inheritance. Let's get into DNA replication. All right, so you can split the process of DNA replication into 3 general steps or phases. That includes initiation. Basically think of it like this. It has to start, right? So it has to initiate. And that occurs at something called the origin of replication. I'll tell you more about that in a moment. Elongation. This is the part where you're extending that new DNA strand. And what does that is the enzyme called DNA polymerase, a very aptly named enzyme. And then, of course, the whole process has to end at some point. So this is where replication is terminated, and we'll see how that happens in different kinds of organisms. All right, so a couple of key features here. DNA synthesis occurs in 2 directions, right? So if we think about that double-stranded nature of DNA, we're going to start or initiate replication at an origin of replication and separate the 2 strands. So now we have 2 template strands that we can make copies of. And then this copying is going to occur bidirectionally, 2 directions. So we see that here in a bacterial chromosome, which is circular. Here's the origin of replication. So the DNA unwinds, separates here, and we see replication going this way to the left and also to the right. So it's, again, bidirectional. We see the new DNA forming that's complementary to the old DNA. In eukaryotic chromosomes, these are much longer. They're not circular. They're linear molecules. There are multiple origins of replication. It would take too long if you just started in one place and copied from there outward. So there are actually multiple origins of replication on a chromosome, so it can get started in multiple places. And when you have this opening, and this ever expanding opening, really, which will ultimately become 2 strands, this is called a replication bubble. It creates this open spot, and we see that here in this electron micrograph. This is a bacterial chromosome being copied, and this opening where you've got the new DNA or the replicated strands, and then this bubble, this open space in between them. All right, so who are our players here? We need a few different proteins and enzymes that are super important. One is a helicase. So a helicase helps unwind, helps separate the 2 DNA strands. That's going to open them up. That allows that replication machinery to get in there. So think of it like a zipper almost. This helps open up that zipper, separating the 2 strands. And remember what it has to do. It's just really breaking those hydrogen bonds that hold the bases together. So it's not a huge task, but it moves along and creates the front end of the replication fork. So here's our bubble, and at either end is a replication fork. Another set of proteins that's important here, this single-stranded DNA that we've created when we open up this bubble is not stable. So it needs these single-stranded binding proteins that help temporarily stabilize the DNA until a copy can be made. And another important enzyme here is this topoisomerase. And you see it just sort of sitting out here like, well, wait, what is it doing just hanging out on this double-stranded part of the DNA? Think of it like this. Say you take 2 strings, and you can do this, wind them together, think like double helix, right? And secure the end, put it under your foot or something like that. And then grab the other ends of the string and just start pulling them outward. It's not just going to simply unwind and, oh great, we got 2 strands now. You'll see it'll get knotted up halfway down. It'll twist even more and get knotted. And so what topoisomerase does, it actually nicks the DNA of one of the strands, and that helps relax that tension on the twisting. So that's very important. All right, the other enzyme here, we'll get to that on the next slide. This is called primase. Because the molecule, the enzyme that makes that new DNA strand, it can't just start anywhere. It needs a 3' hydroxyl. Remember, think back to the end of a DNA strand. It has that it's the 3' end, and it has a hydroxyl group. DNA polymerase needs that as a place to start building. And so if you just have a single-stranded DNA polymerase, it can't start anywhere. So what living things do is they first build a little piece of RNA. It's complementary to the DNA bases, and that will provide that 3' hydroxyl for DNA polymerase to work from. And the enzyme that builds that little RNA primer, it's called primase. And so these are about 5 to 10 nucleotides long, and they're temporary, as we'll see. All right, so here's where we can look a little closer now. Here's one of our strands in blue, highlighted in blue, the sugar phosphate backbone. This is oriented 5 to 3' going up. If we're building a new strand, it has to be anti-parallel, so it's going to be 5 to 3' going the other direction. And here's that 3' hydroxyl where new nucleotides are added by DNA polymerase. And if we're talking about prokaryotes, where we learned initially a lot of this stuff, it's specifically DNA polymerase 3 that does that job. There's a few other DNA polymerases as well. And so we add to that existing chain. It's now 1 nucleotide longer. Here go 2 extra phosphates from our incoming nucleotide. That's a pyrophosphate group, just these 2 phosphates together. And so we've extended that chain. So here it is again, just seeing it in a different way, using a different model here. Again, pay attention to the 5' to 3' template. Our new strand is oriented the opposite direction. We have complementary base pairing. Here comes a thymine to match up with this adenine here. And here is DNA polymerase. Again, DNA polymerase 3 if we're talking about E. coli. And these pyrophosphates peel off here. Okay, so let me ask you this. Is there any difference between ATP added to this DNA strand and that energy currency of the cell, ATP? All right, yeah, there is a difference. That ATP, that's the energy currency of the cell, is an RNA nucleotide. But other than that, no, they're pretty much the same, except, that the ATP as the energy currency has that ribose sugar. That could be used to make RNA, though, as we'll see later when we get into transcription. Now, it's worthwhile mentioning, when we think about how a strand of DNA grows, we're always adding to that 3' end. And you might ask, well, why? Why can't we build DNA strands the opposite direction? It has a lot to do with those nucleotides being kind of providing the energy, some of the energy for that reaction. Again, think like ATP, energy currency of the cell. Well, deoxyribonucleotides can provide that energy as well with their triphosphates. And so let's look at a couple of scenarios, whether we're adding to one end or the other. Let's look what actually happens. So that's the example on the right of this figure. So here's our triphosphate nucleotide, and it's going to be added to the end here. So we see that happening. We break off these pyrophosphates, the 2 phosphates here, and that provides the energy for this. So if a mistake is made, DNA polymerase has a proofreading ability, and it will chop off this mistaken nucleotide. Great. And okay, we need this other nucleotide to come in. No problem. We still have the energy because each new nucleotide brings in that energy. On their 5' end, they have these phosphates. And so, okay, no problem, add a nucleotide. So that's what actually happens, and we think that this is why this evolved. It's because of this proofreading. So why doesn't the opposite happen? Think of it like this. Here's our growing strand. And on our 5' end of our actual strand, that's where our phosphates are. So this is where our new nucleotide is going to attach. So in comes our nucleotide. The phosphates peel off from the strand, not from our nucleotide. Now, if we look here, that seems fine, right? We got a nucleotide attached. We still have phosphates. Great. Bring in the next nucleotide. The problem happens here. If we need to now edit that nucleotide out, a mistake was made, DNA polymerase proofreads it off. Now we have our strand that lacks those triphosphates. We have a strand that has the 1 phosphate. We don't have the energy needed to add it. Sure, the next nucleotide has 3 phosphates, but where it's binding, that's not where the 3 phosphates are. These need to provide the next, would need to provide the phosphates for the next nucleotide, not this one. So this is why the DNA synthesis has evolved in this manner, because of energetics. All right, so we're looking at elongation. How do these new strands get made? Elongated. And we have 2 things that are happening here in our replication bubble. Well, it's going bidirectional, so say left and right. And on one of our strands, the new strand can be synthesized continuously. This is called the leading strand because the new strand is being formed 5' to 3', again, anti-parallel to the template, which runs the opposite direction. That's something when you're first learning about this, you want to really have a good handle on that. Whatever scenario you're thinking of is that you end up with anti-parallel strands. So our new strand being built 5' to 3', here's our little RNA primer. DNA polymerase is extending this, and it can just happily cruise along and add nucleotides as that replication fork continues along. Where the problem comes in is that other strand, because it's oriented in the opposite direction, our other template strand. So here it is, we're opening our replication bubble. We can't just follow the replication bubble because our new strand can't be built 3' to 5'. It has to be built 5 to 3, really back toward our origin of replication. And so this is called the lagging strand because it takes longer to replicate because you need to do these things here. So as the bubble opens up, an RNA primer is formed just like we see anywhere, and in comes DNA polymerase 3, and fills up to wherever the next RNA primer is. And this little bit of stretch of DNA is called an Okazaki fragment. And as the replication bubble expands, you get the next Okazaki fragment, and the next one, and the next one. And this is just backfilling this space, working in the opposite direction. All right, so now we're stuck with a bunch of little RNA primers. So what do we need to happen? Well, this DNA polymerase 1, this comes in and rips out those RNA primers and fills in with DNA. And then something called DNA ligase. It ligates, connects those Okazaki fragments to make a continuous strand. So ultimately, everything is still moving to the left here in this part, on this side. Of course, the same thing's happening over on the right here as well. So ultimately, we're continuing replication this way. It's just that the leading strand can follow. The lagging strand has to fill 1, and then fill 2, then fill 3, and so on as it goes. So a few additional details on the lagging strand. Again, it's working away from the replication fork. Those bits that are being synthesized are called Okazaki fragments. And the length of those varies depending on the species. In eukaryotes, they're smaller chunks, about 100 to 200 nucleotides long. In prokaryotes, like E. coli, you can get longer stretches before it has to make a new Okazaki fragment. And then again, DNA polymerase 1 fills in those RNA primers, and DNA ligase seals things up. And so here you can see the whole thing kind of in action, a little zoomed in. And check out this video. I think this is a handy one to look at, and there's all sorts of resources online to see how this is working. Okay, so during DNA replication, which enzyme removes those RNA nucleotides and adds DNA nucleotides to the Okazaki fragments? So you had a couple of probably enticing choices with 2 different kinds of DNA polymerase, and it's that DNA polymerase 1 in E. coli that does that backfilling. Now, we're looking at this very linearly. So we go back to this figure here. We're seeing that replication fork and the leading strand and the lagging strand. Just, for your own interest here, this is actually what's happening. So if we model what's going on here, this helicase, remember that's unzipping the DNA strand, it is connected to the DNA polymerases. So here's this leading DNA polymerase 3 following the replication fork right here. Meanwhile, the other DNA polymerase that's working on the lagging strand, it's also connected. So how can that be, right? Because it has to backfill over here. The strand twists around, or loops, I should say, loops around. So this is called the trombone model because you have this long strand that loops out this way. And here's that RNA primer and the DNA polymerase working, again, 5' to 3'. And the reason that works is the strand is twisted around the other direction. All right, how about rates of synthesis? So how fast does this work? So generally, obviously, every organism might be slightly different, but generally, bacteria, prokaryotes, they'll do about 500 to 1,000 nucleotides per second, pretty good. Mammals, about 50 nucleotides per second, so a lot more slow. There's more proofreading that occurs in mammals, and you think about the genome is just much larger, and they're wrapped in remember the DNA is wrapped in histones in eukaryotes as well, so that's going to be slower. Now, eukaryote genomes are also typically bigger too, so not only is replication a little slower, but 50 per second isn't anything too bad, but much slower than a prokaryote. They also have much bigger genomes to copy. All right, so what if DNA polymerase makes a mistake? Well, it has its own proofreading mechanism. So here we see this example. DNA polymerase here added an incorrect base. So here it should be a guanine, not an adenine base. And so that can be removed. So DNA polymerase has this exonuclease activity. Exo basically means the end. It'll take nucleotides off the end of a strand. And so here it is removing this mistaken adenine, and now it can fill in with the correct base. How often do mistakes occur? Again, depends on the species and the polymerase, but approximately 1 in 10 billion. So not very often. And not all of your genome is actual genes. There's a lot of stuff that's in between the genes. So if a mistake occurs there, it's probably not a big deal. But if a mistake happens, it can be corrected, except 1 in 10 billion times it's not. Now, if DNA polymerase misses something, keeps reading through it, doesn't proofread a mistake out, that's not the end of the world. So something called mismatch repair can occur. So here we see the mismatch. This should be an adenine, not a guanine. And there's a variety of genes involved in this, but here we see these enzymes clipping the sugar phosphate backbone, and then an exonuclease, starting at that loose end there, works its way back and trims out several nucleotides. I was going to say all, but not all of them. Several nucleotides. Now another DNA polymerase can come and backfill this and the correct nucleotide inserted. There's other reasons mismatches might occur. So chemical damage, or X-rays, for example, or UV damage, as we see here. So we're seeing this example here where this DNA is hit with UV, and instead of these nucleotides base pairing across the strand, they start pairing with each other on the same strand, and so that causes a problem. So these mistakes or issues can be fixed by a couple of these other mechanisms. So one is called base excision repair. And in this case, it's kind of interesting because let's take the example here. A uracil is inserted into a DNA strand. Now, that's not supposed to happen because there shouldn't be uracil in DNA. But cytosine, which should go here, is actually very similar in structure to uracil. So you could see it happening. But what happens here, the base excision, first, the base itself is removed. Remember, the nucleotide is made out of the sugar and the base and a phosphate, not shown here. But we have the base actually just clipped out. Now, then the rest of that sugar phosphate backbone has to be, that piece has to be removed, and then the correct base comes in. Nucleotide excision repair, this actually looks like a lot like our mismatch repair. So here's that case where nucleotides are binding to one another on the same strand. This is called a thymine dimer in this particular example. And so what happens here? This is detected, this is a problem, and a variety of enzymes come in, clip that sugar phosphate backbone in two places, and then remove that chunk, this whole piece, get it out of there. And then DNA polymerase comes in and fills that in. And then finally, this enzyme called DNA ligase, which we saw in DNA synthesis before, it seals those little gaps in the sugar phosphate backbone. All right, let's look now at that last phase of DNA replication, which would be termination. So the end of it has to end somewhere, right? So If we're looking at a bacterial chromosome, which is circular, it's pretty easy. So what's happening here? Here's the origin of replication, usually only one in a prokaryote. Here's our replication bubble, our replosome, that's the DNA polymerase on the leading and lagging strand together going one direction. Here's another one going the other direction. Eventually, they're going to run into each other. And that's where the synthesis terminates. Now you have two circular chromosomes as a consequence of that. How about eukaryotes? Well, we know there are multiple origins of replication. And so eventually these replication bubbles or replication forks are going to run into each other. So here we had two. As they get bigger, they eventually merge, forming this larger replication bubble. Now, as the replication forks work their way out to the ends of the chromosomes, they'll just run off the end, right? So that's how that's going to terminate on the ends. And the ends of the chromosomes are called telomeres. Telo means end. And these are really important for a couple of reasons. Let's have a look what's happening in DNA replication. The leading strand, that can just be synthesized right to the very end. And again, that complex, polymerase complex, can just run right off the end. And you can go right to the very end of your template strand. The issue here is on the lagging strand. Remember, we're always backfilling. So this Okazaki fragment gets made, then this one gets made, then this one gets made. And we always need an RNA primer. So here we're going to zoom in on the very end of our telomere here. Here's our RNA primer. And we've backfilled the DNA here. Well, the RNA primer is removed, but now what? How do we fill this in? Because remember, DNA polymerase needs a 3' hydroxyl. There's none here. There's nowhere to put another primer. And so we've lost a little bit off the end of this chromosome. And so when, say, the next round of replication happens, this light blue one will be a template here. And here, this will be the new leading strand complementary to that. It can go to the end. But this is shorter now. So if we have multiple rounds of replication, so every time the chromosome is copied, it gets a little bit shorter. As we see here, subsequent rounds of replication get shorter and shorter. All right, so this is in eukaryotes where we find telomeres, and we have kind of this issue. In humans, there's this repetitive sequence of these 6 nucleotides, hundreds, thousands of these repeats. In other organisms, that repeat sequence is something different. And so this allows for kind of a buffer on the ends of the chromosomes, because if this continues to happen, right, you're always losing a little bit of your chromosome every time it's copied, eventually that's going to reach where genes are and start to eat away at those. And so that would not be great. All right. Now, you don't have to worry, though, that over many generations of humans, we're all going to have shorter and shorter chromosomes and start to lose genes, because something happens in specific cells, so listed here, germline cells, those that produce gametes, some stem cells, and interestingly, cancer cells. They have the activity, or there's an enzyme that's active in those cells called telomerase. So that's the enzyme A's, right? It works on telomeres. So here's what telomerase is doing. It's adding additional repeats on this one strand. It's adding here the TTA, GGG, that's the human repeat. It's adding more of those on the end. Now a little RNA primer comes in. It can start here and basically add to the end of our chromosome instead of losing it. So this is important, obviously, in germline cells. So the gametes that fuse to form a new individual, a zygote, that kind of resets the clock for that individual in terms of those chromosome length. This is an area of real interest in looking at aging. So there's been a lot of interest in this, thinking, well, can you almost reverse aging of cells by encouraging the telomerase activity in other cells, kind of resetting the clock, as it were, on these chromosomes? Okay, here's a review type question. And now that we're wrapping up DNA replication, which following sets of materials is required for both eukaryotes and prokaryotes? All right, this next part is all about genetic engineering. So we understand DNA replication, we understand DNA structure. We can now use that in genetic engineering and manipulating DNA. And so there's a few things to kind of pay attention to as we talk about this. This idea of nucleic acid hybridization. Now, that's not as complicated as it maybe might sound at first glance. All that is about is that if you had a DNA strand, like I show you at the bottom here, here it's 5' to 3', you could create another DNA strand that's complementary to it. Here we're showing it in reverse complement, 5 to 3 going right to left. This will base pair with our target DNA strand. Here it matches up, right? So, and therefore it will hybridize, it will stick to that spot. So this is really important, is using this idea or property rather of nucleic acid hybridization because of the base pairing for genetic engineering. So this is really foundational in the next few things that we're going to talk about. There are a few big advances that we'll cover here. The discovery of restriction enzymes, you'll hear about what those are. DNA cloning, we'll start there. And then the polymerase chain reaction. Now there's other important advances too, but these are just fundamental, and most any lab that does some molecular biology probably uses these three things on a regular basis. So let's start with cloning. What does cloning mean? Well, it means you're making copies of usually a gene, that's what we're often interested in, or maybe some other kind of DNA fragment that we're interested in. And we want to have a way to make many, many copies of it. So we have a lot to work with for whatever experiment that we're interested in. We can isolate it from other fragments, etc. And so something we take advantage of here is that bacteria, many bacteria, have these extra bits of DNA called plasmids. So here's our bacterial DNA, their chromosome, it's much bigger. And then you have these small extra chromosomal bits of DNA called plasmids. And so we can use those, take them out, put genes in them, manipulate them, and the bacteria will also take those up. So getting them back into the bacterium is reasonably easy. And so this allows us to manipulate DNA fairly easily. So what are we doing when we're cloning? We're combining a plasmid usually and DNA from some other source. We say foreign DNA, but it's some other source, foreign to, say, that bacterium. And we create recombinant DNA. It's a combination of these two things, the bacterial plasmid DNA and then a chunk inserted into it of the foreign DNA. The next thing we want to do is get it back into the bacterium. Now remember Griffith's experiments with Streptococcus pneumoniae in mice? He knew there was some transforming factor. Bacteria were picking up DNA from their environment, from those heat-killed bacteria. Well, we can do that here too. We create our recombinant DNA construct, and then we mix it with bacteria that will take it up and then express that gene or just copy it. And then there are a myriad applications of this sort of thing. Agriculture, medicine, genetics development. Say an example from medicine. Anybody, or if you know somebody who is, say, diabetic and takes insulin, that insulin is being produced by bacteria. It's a human gene that's put into, say, E. coli, that then expresses that protein, and then we can purify it. Now we have loads of insulin at hand. All right, so that example of insulin is a case where we're targeting a single gene. And so that's often what we're doing. And this is called gene cloning. And we're putting it into a plasmid. The plasmid is the cloning vector. So you think of it, a vector is something that carries something else. So the plasmid's carrying it here. Again, they're joined together, but the plasmid is the bigger partner. It allows us to make many, many copies of it. Or again, produce a protein that we can then harvest like we see here. So very, very useful. So that's the basics of DNA cloning. We now can look at introducing this idea of restriction endonucleases. So what are these things? They're enzymes. And so what they do is they'll cut DNA at very specific sites. They have a recognition sequence. They'll only cut at that specific sequence of bases. Many of them will leave what are called sticky ends, little overhangs. We'll see how that works shortly. Then you can, cut two different pieces of DNA, a plasmid and some other gene of interest from somewhere else, mix them together. That's that idea of making recombinant DNA. And because they have the same sticky ends, they will stick together by base pairing. Remember, that idea of hybridization is critical here. And then we need to join the sugar phosphate backbones. That's done by DNA ligase. Remember DNA ligase from DNA replication. We can use it here too in the lab. So these restriction enzymes that we use all the time in genetic engineering, they're technically type 2 restriction endonucleases, or double-stranded endodeoxyribonucleases. Again, sounds like a lot, but we can break it down into pieces here. Restriction endonucleases, I'll tell you why that we call them that in a second. Double-stranded, okay, we're dealing with DNA, it's double-stranded. Endo, that means it's cutting within the strand somewhere, not out at the very end. That would be an exonuclease, but we're interested in cutting our strand of DNA somewhere along its length. And then deoxyribo, that we're talking about DNA here, and nucleases means it attacks nucleic acid. All right, so the discovery of these things is really interesting because these were discovered in bacteria as a defense mechanism. So remember our bacteriophage here injecting its DNA. Bacteria produce these enzymes to chew up viral DNA. So this is where this term restriction comes from, because these enzymes restrict the host range of bacteriophages. So some bacteria have these and they'll prevent an infection, and that restricts that range. So, well, why don't these enzymes just chew up the bacterium's own DNA? How come that just doesn't get destroyed as well? Because bacteria can do something called methylation, and this will add methyl groups to their own DNA. And the restriction enzymes, they will not attack those methylated sites. So that will protect their own genome. Meanwhile, the enzymes will take apart these viral genomes. All right, let's circle back to cloning now that we know something about restriction enzymes. So here's a plasmid. This is going to be our vector. And we use a restriction enzyme. It's going to cut at this very specific sequence, and it will cut unevenly. This is this idea of creating sticky ends. So one strand it'll cut here between the G and the A, and the other strand, it's actually also cutting between the G and the A on the other side here, and it leaves these overhangs, and they're called sticky ends. Now we take a fragment of interest, some other gene from something else, that's that foreign DNA, with the same sticky ends, and so they overlap. Then we can seal that up with DNA ligase, and now we have this vector and our foreign DNA. That's our recombinant plasmid. We can look a little more closely at these sticky ends. So here is this one enzyme, EcoR1. It has this recognition site. I'll tell you more about that in a second. Leaving these overhangs. Here's another enzyme that cuts at a different restriction site, and it leaves different overhangs. Now look at these sequences. So we can go 5' to 3' here, GAATTC. And then remember, the other strand is anti-parallel, so we'd actually start on the other end here, GAATTC. It's the same. So these are called palindromic DNA sequences. If you think of a palindrome in English, that's something that if you took the spelling of it forward and backward, it's going to give you the same thing. So taco cat, for example, you flip those letters around, it still says taco cat. Now, it's not quite the same thing here because we're actually dealing with two strands. But the idea here is when we read this one strand, it's going to say the same thing as the other strand does the opposite direction. And so that's why we call it these palindromic DNA. All right, so here's a challenge for you. Which of these is not a palindromic DNA sequence, given what you just learned? All right, yeah, it's B, because if we read this strand this way, AAGG, this other strand doesn't read AAGG, it's TT. So that doesn't work. The rest of them follow that palindromic, DNA palindromic pattern. All right, so let's look at these sticky ends here again. So we've produced these sticky ends on 2 different fragments. And if we look close, as these come into close association, the adenines are going to base pair with the thymines. Again, think back to that first slide when we talked about genetic engineering, that hybridization is just super critical. So these are going to be drawn to each other because they complement one another. And then once they get close enough, showing these blue lines here, we see DNA ligase will help join that sugar phosphate backbone, so it becomes a continuous strand here and the other strand also continuous. So there's all kinds of restriction enzymes. Here's a list from a company showing the ones that they offer. And so they're really useful. And you can find a certain DNA recognition site. You might be able to find an enzyme for it. They're so varied. And so this is really useful in the lab. All right, so we've got our recombinant DNA now, and then let's go back to this idea of transformation. We're going to mix it with, say, E. coli, a bacterium, and that bacterium will pick this up. Sometimes we need to encourage them chemically or using electric current. They'll take those up more readily. And so now this plasmid is inside the bacterium. Now, what prevents this from just getting kicked out? The bacterium replicates, and whether it has the plasmid or not, I mean, who cares, right? It's got the genes it needs in its genome. So one way we can do this is to make sure that our vector, our plasmid, has an antibiotic resistant gene. What that's going to do, it's going to produce an enzyme that confers resistance to some antibiotic, let's say kanamycin. Now when we grow this bacterium on a medium that contains kanamycin, the only way a bacterium can grow is if it's picked up at this plasmid. And they're not going to lose the plasmid. Any bacterium that loses the plasmid will die in the presence of that kanamycin. So this antibiotic resistance is super important. And we can then replicate our bacterium. We used to have one bacterium with one clone in it. We can replicate these overnight, and then we have millions or billions of copies of this. So transforming bacteria is pretty straightforward. Transforming eukaryotes is much more difficult, right? With different genome system, how they take up foreign material is very different. So here's just a couple of examples of things that we can do. Microinjection, actually injecting a DNA sample into the nucleus of a cell. We actually can use viral vectors. Some viruses actually move into the nucleus of eukaryotic cells, and that could be a way, or is a way, that we can introduce or transform eukaryotic cells. Another really useful technique we use is a way to separate out DNA fragments based on their size, and this allows us to visualize, say, what DNA fragments we have in a sample that we've prepared. This is called electrophoresis. The electro part has to do with applying a charge to these DNA molecules, or putting them in a system where there is an electric current. And DNA is negatively charged, so it will migrate towards this positive electrode. Now, in water, let's say we just mix DNA in water and apply a charge to it, regardless of size, DNA is going to move toward the positive electrode. But if we put them into, say, like a gel-like matrix, they will separate by size. The littler ones are going to migrate farther because they're small. They can weave their way through that matrix more quickly than the bigger fragments. So this allows us to separate these out. And so we can see that here where we would load our sample in these wells in this sort of block of gel-like substance, apply electric current. The smaller stuff will migrate further, the bigger stuff won't migrate as far, and it allows us to see these different fragments. So here's a well here, and this is a ladder, a marker with known fragment sizes. And so we have, say, let's say this is 100 nucleotides, 200, 300, and then we got to jump here. Maybe this is 600, 700, 800, etc. And so the further to the left on this example, the bigger the fragment is. And here are some samples that we loaded up that were, say, cut with a restriction enzyme. And we've produced fragments of various sizes, small, medium, larger. And if we want to know how big they are, we can compare them to this marker that's in this top and bottom lane where we have known fragment sizes. So we can figure out what we have based on the relative migration of those known fragments. All right, now you try. So here's a DNA fragment. It's cut with restriction enzymes 1 and 2, producing these 3 different chunks of DNA. If we run them on a gel electrophoresis rig, what pattern would we get, given your choices here? All right, so you've got to pay attention to the positive electrode and the idea that smaller things are going to get further along toward the positive electrode than bigger things. So C will be the biggest fragment, so it should be the furthest to the left. So we can cross off A and C as options here, and we're left with B and D. And the fragment order is the same here. But if we look at D, we have fragments that maybe look about the same difference in size from one another, but that's not what we really have here. C is really big, B is tiny, A is maybe twice as big as C, and so we can go with B here based on that. So we're kind of down to B and D, but given the relative sizes, B is the best option. All right, let's finish up genetic engineering, talking about this technique called PCR, the polymerase chain reaction. This is probably one of the most important tools and techniques that we use in molecular biology. Even if you're not a molecular biologist, you'll probably do PCR at some point. We use it for a huge range of applications, and it involves the amplification of a fragment of DNA, a target fragment of DNA in, say, a genome. And this is the example I use. The genome's really big. We don't want all that genome. We just want a little bit of it. And we want to make a lot of copies of it so we can do something with it. And so PCR is really useful if, say, we don't know what the sequence of a gene is we're after, but we can target it based on other known organisms. If an organism is unculturable, like a bacterium or a eukaryote or whatever it happens to be, we can find it using DNA detection methods, using PCR. If it's there, we get amplification and we say, oh, there it is. You can start with really tiny amounts. So this is great as a diagnostic tool. So think about what's going on with coronavirus outbreak. The diagnostic test uses, or one of the diagnostic tests uses PCR, so it can amplify very little starting material and get a detection there. So it can be also very specific depending on how it's designed. So you can only target that gene of interest, or maybe you're targeting a set of genes. You can be a little less specific if you like. All of it relies on this basic idea. If we have DNA that's held together by base pairing, and those are hydrogen bonds, if you heat it up, it will come apart. And we call this melting when the strands separate. And then if you cool it down, those strands will come back together, or complementary parts of DNA. Remember back, hybridization, super important. These will rehybridize and form that double-stranded molecule again. Heat it up, we can just keep going around and round. So this heating and cooling is really useful, but just heating up a DNA strand and cooling it off again isn't that helpful. What we do next is we actually add little bits of DNA that are a known sequence that match up to regions flanking the gene that we're interested in. And so we see that here. We heat up the DNA, that's shown in green, so now we have a single strand. And now we're going to cool things down a little bit, and it allows this, what we call a primer, to stick to the complementary region. Now that this gives DNA polymerase a place to start. Remember, DNA polymerase needs a 3' hydroxyl, and it makes a copy. So it extends it at, we'll see, 72 degrees approximately. And so we can do this over and over. So we'll melt the DNA, separates the strand. We cool it a little bit. That allows our little primers to bind, flanking the region of interest, but not so cool that the both big strands come back together. DNA polymerase will extend those strands. Now we have 2 copies. Now we can melt those, and we can go round and round and round. So this was invented, this technique, PCR, by this guy, Kary Mullis. Very interesting character. Go check him out, Google him, and read some of the interesting, bizarre, sketchy things about him. But he won the Nobel Prize for Chemistry in 1993 for basically putting this all together. Now, people were able to do this already, copy a little bit of DNA, but one of the challenges was, well, remember, you have to heat up the strands to separate them. And every time you heat up those strands to separate them, you kill your DNA polymerase. Because say it's E. coli DNA polymerase, it doesn't work at 95 degrees Celsius. It gets destroyed. So what Kary Mullis put together here was taking this DNA polymerase from Thermus aquaticus. This is a bacterium that lives in hot springs. So the enzyme works great at high temperatures, and it doesn't get destroyed if it's heated up to 95 degrees. And so you can do this PCR with a little primers binding, heat it up, or replicate, heat it up, and around we go, and without having to add more DNA polymerase every time you heat up your sample. And so you need these four things. You need your template target DNA. You need those primers that are specific to the region you're interested in. You need those building blocks, the nucleic acids that are going to be added as you're making new strands. So your ATP, CTP, GTP, TTP. And then you need that enzyme, Taq DNA polymerase, that can withstand the heat. And here's that cycling. So melt, separate the strands, primers bind, and then make a copy. Melt, primers bind, make a copy, etc. And so effectively, every time you make a copy of one template, you're doubling at every time you go around that cycle. And so let's say at 3 cycles, so we use 2 to the n copies, right? So 3 cycles is 2 to the 3, you'd get 8. So if you started theoretically with 1, you'd end up with 8. And that's shown here in this example from your text. Usually in PCR, we'd run about 35 cycles. So a theoretical yield here is 2 to the 35, which is 34 billion. So this is an amazingly useful way to get a lot of a specific region of DNA that you can then do something with. Okay, so we'll finish here by, again, coming back to cloning as we started when we talked about genetic engineering. Guess what? If you use PCR to make billions of copies of a fragment of your interest, you can plug that into a cloning vector, a bacterial plasmid shown here. So here's our DNA fragment from PCR. Maybe we have to cut it to get the sticky ends. There's actually many ways to get these into plasmids. And we ligate in that fragment. Now we have our recombinant DNA, yet another application of PCR, and yet another way we can see how cloning is so very useful. Okay, that's it for this chapter. See you next time.